Have you ever wondered if it's possible to edit the very fabric of life itself? Imagine a world where we could rewrite our genetic code, just like editing a piece of text. Welcome to the revolutionary scientific breakthrough known as gene editing technology. This isn't the stuff of science fiction anymore. It's real, and it holds the potential to change our world in ways we can barely begin to comprehend. So buckle up, because we're about to embark on an extraordinary journey. Welcome to the world of gene editing technology. So, what exactly is gene editing technology? Imagine life as a grand symphony, with each instrument playing its own unique part. Now, think of genes as the sheet music that guides each instrument. Genes are essentially the instructions for life. They determine the color of your eyes, the texture of your hair, and even the shape of your nose. They are small segments of DNA, the molecule that carries the genetic blueprint of all living creatures. In each of our cells, tens of thousands of genes work together, orchestrating the complex performance that we call life. But what if one of those genes is out of tune, causing a genetic disease or disorder? This is where gene editing technology steps in to correct the discordant notes. Gene editing technology is a revolutionary tool that allows scientists to manipulate the genes within an organism. Imagine being able to rewrite the sheet music, changing the notes to create a different melody. That's essentially what gene editing does. It uses a kind of molecular scissors to cut out, replace, or insert specific genes within the DNA sequence. Traditional genetic engineering methods are like trying to play the piano with boxing gloves on. They can make broad changes but lack precision. Gene editing technology, on the other hand, allows for precise, targeted changes to the DNA sequence. It's like swapping out the boxing gloves for a conductor's baton, offering an unprecedented level of control. This technology could open the door to treating or even curing genetic diseases, enhancing crops, fighting off pests, and so much more. The possibilities are as vast as the genetic code itself. But remember, with great power comes great responsibility. Just as a conductor must carefully guide the orchestra to create a harmonious symphony, so too must scientists use this technology wisely to ensure the best outcomes. In essence, gene editing technology allows scientists to alter these genes, changing the course of life itself. Now that we know what gene editing technology is, how does it work? Well, let's think of our genes as an intricate and complex book of life. The book is written in a language of 4 letters, A, T, C, and G, representing the 4 nucleotides that make up our DNA. Sometimes, a mistake in this book can lead to diseases. That's where gene editing technology comes in, acting as a proofreader and editor, fixing the mistakes or introducing new elements into the story. But how does this editing process actually work? It all starts with a class of proteins known as enzymes. Enzymes are the workhorses of our cells, performing countless tasks, including the cutting and pasting of DNA. In the context of gene editing, certain enzymes known as endonucleases are employed. These molecular scissors can cut DNA at precise locations, allowing scientists to remove, add, or replace specific genes. One of the most popular and widely used gene editing tools is CRISPR-Cas9, a revolutionary technology that has taken the scientific world by storm. The name CRISPR-Cas9 might sound like something out of a sci-fi novel, but it's actually an abbreviation for clustered regularly interspaced short palindromic repeats and CRISPR-associated protein 9. So how does CRISPR-Cas9 work? Imagine you're looking for a specific word in a book. You could go through the pages one by one, but that would take forever. Instead, you use the search function in a digital version of the book to find the exact page and line you're looking for. That's essentially what CRISPR does. It uses a piece of RNA, a close cousin of DNA, as a guide to find the exact location of the gene that needs editing. Once the target gene is located, the Cas9 protein acts as the molecular scissors, cutting the DNA at the exact spot. Now we have an open space in the DNA sequence. This is where the magic happens. Scientists can insert a new gene or correct the existing one in this open space. The cell's natural repair machinery then stitches the DNA back together, completing the editing process. But it's crucial to remember that gene editing is not as simple as it might sound. It involves a complex and delicate process, requiring precision and control. A single mistake can have far-reaching consequences, potentially introducing new diseases or other unforeseen problems. Therefore, while gene editing holds immense potential, it must be approached with caution and respect. Just like a word processor, gene editing technology can cut, paste, and replace genes, but the canvas here is life itself. The question now is, what can we achieve with gene editing technology? Just imagine a world where diseases are no longer a death sentence, but a problem to be solved. This could be our reality with gene editing. Many of the diseases that plague humanity are genetic in nature. These include various cancers, cystic fibrosis, Huntington's disease, and muscular dystrophy. By precisely editing the genes that cause these conditions, we could potentially eradicate them from our genetic lineage, saving future generations from their devastating impacts. It's like having a molecular scalpel that can cut out the diseased portions of our genes and replace them with healthy ones. But the potential of gene editing extends beyond human health. In agriculture, for instance, gene editing technology could revolutionize the way we grow our food. Imagine crops that can withstand harsh weather conditions, resist diseases and pests, or even be nutritionally enhanced to address specific dietary needs. By modifying the genetic makeup of plants, we could potentially increase crop yields, reduce the use of harmful pesticides, and help address the global food security issues. It's like having a toolbox that allows us to design our food according to our needs and the planet's needs. And what if I told you that gene editing could potentially extend your lifespan? Some scientists are exploring the idea of using gene editing to slow down or even reverse the aging process. The key lies in our telomeres, the protective caps at the ends of our chromosomes that shorten as we age. By tweaking the genes that control telomere length, we might be able to slow down the aging clock, allowing us to live healthier and possibly longer lives. It's like having a fountain of youth at the molecular level. But let's not forget about the potential applications of gene editing in the animal kingdom. Scientists are already using gene editing to create animals with desirable traits, such as pigs that are resistant to swine flu or mosquitoes that can't transmit malaria. This could lead to significant improvements in animal health and welfare, as well as help control the spread of diseases from animals to humans. Of course, all these potential applications come with their own set of challenges and ethical considerations, which we'll delve into in the next scene. But one thing is clear: gene editing technology holds immense potential. It's a powerful tool that, if used responsibly and ethically, could transform our world in ways we can barely imagine. So, let's dream big, let's dream responsibly, and let's push the boundaries of what's possible, while always respecting the sanctity of life and the natural world. Because at the end of the day, the possibilities with gene editing technology are limited only by our imagination and ethical boundaries. Yet with great power comes great responsibility. And so it is with gene editing technology. This revolutionary science is not without its ethical quandaries. The power to alter life's blueprint can be a double-edged sword, with potential for both unprecedented good and unimaginable harm. At the heart of the ethical conundrum is the question of consent. When we edit the genes of an unborn child, we are making decisions that will affect them for their entire life. They have no say in the matter. Is it right to make such profound decisions on their behalf? It's a question that's sparked much debate among ethicists, scientists, and the public alike. And then there's the issue of equity. If only the wealthy can afford gene editing, it could create a genetic upper class, widening the gap between the haves and the have-nots. Imagine a world where the rich not only have more wealth, but also superior health, intelligence, and longevity. This is a potential future we must strive to avoid. Moreover, the prospect of designer babies is a hotly contested topic. Should parents be allowed to choose their child's physical traits, such as eye color, height, or even potentially their intelligence? While some argue this is no different than selecting a school for a child's education, others contend it's a step too far, a step into playing God. There are also concerns about the unintended consequences of gene editing. Even with the most meticulous planning and execution, we cannot foresee all the potential ramifications of altering our genetic code. An edit intended to prevent a disease could inadvertently introduce new vulnerabilities or health issues. And let's not forget the potential for misuse. In the wrong hands, gene editing technology could be exploited for nefarious purposes, such as creating biological weapons or genetically enhanced soldiers. It's a chilling thought that underscores the urgent need for robust regulation and oversight. Yet despite these ethical dilemmas, the potential benefits of gene editing are hard to ignore. The prospect of eradicating genetic diseases, enhancing crop yields, and even reversing the aging process are tantalizingly within our grasp. The key, therefore, is to navigate this ethical minefield with care. We must establish clear guidelines and regulations to ensure that gene editing is used responsibly and ethically. This includes rigorous scientific oversight, transparent public dialogue, and equitable access to the technology. We must also foster a culture of humility and caution. We are, after all, tinkering with the very fabric of life. We need to respect the complexity and interconnectedness of our genetic code and be mindful of the potential for unintended consequences. In conclusion, gene editing technology holds great promise, but it's not a panacea. It's a tool, and like any tool, it can be used for good or ill. It's up to us to ensure that it's employed wisely and ethically. While gene editing technology has immense potential, it's crucial that we tread carefully to prevent misuse. So, where is gene editing technology taking us? As we peer into the future, we find ourselves on the precipice of breakthroughs that could transform our lives and the world around us. The potential advancements in gene editing are staggering. Imagine a world where genetic diseases are eradicated, where our crops are more resilient, and our livestock healthier. Picture a future where we could potentially slow down the aging process, enhance our physical abilities, or even alter our own genetic code to better adapt to our changing environment. This is not the realm of science fiction, but the potential reality of gene editing technology. In the sphere of healthcare, gene editing could lead to personalized medicine, tailoring treatments to our unique genetic makeup. We could see the development of designer babies, where parents can choose specific traits for their unborn children. However, with great power comes great responsibility. The challenges we face are not just technical, but ethical and societal. How do we ensure the equitable distribution of these advancements? How do we prevent the misuse of this technology? How do we navigate the moral and ethical minefield of editing the genetic code of future generations? The road ahead is not without its obstacles. The science is complex and our understanding is still evolving. But the potential rewards are too great to ignore. As we continue to refine this technology, we must also grapple with the ethical implications and ensure that our decisions are guided by a robust framework of checks and balances. In the face of these challenges, scientists, policymakers, and society at large must work together to navigate this uncharted territory. The dialogue must be open, inclusive, and based on sound scientific evidence. We are at a crossroads. The decisions we make today will shape the future of our species and our planet. As we stand on the brink of a new era, gene editing technology could well shape the future of humanity. Before we end, let's go over what we've learned today. We started our journey with the dawn of a new age, where we delved into the concept of gene editing technology. This groundbreaking advancement allows us to alter the DNA of living organisms, essentially giving us the power to edit life's building blocks. We then unraveled the mystery behind this technology, diving deep into the mechanisms that make this scientific wonder possible. We learned about the process of cutting, copying, and pasting DNA sequences, and we discovered how these intricate procedures can lead to substantial changes in an organism's genetic makeup. From there, we explored the vast potential of gene editing. We imagined a world where we could potentially eradicate genetic diseases, improve crop yields, and even bring extinct species back to life. But with great power comes great responsibility. We reflected on the ethical conundrum surrounding gene editing, discussing the moral implications of altering life and the potential for misuse. Finally, we look towards the future, considering the many ways this technology could revolutionize our world. From healthcare to agriculture to conservation, the possibilities are truly endless. That's gene editing technology in a nutshell, a powerful tool that holds the potential to change life as we know it.